going to get started, although someone reminded me that MIT time says the hour starts at 11:05, and I'm looking at the clock saying 11:02, and I'm thinking that's so weird for MIT. Not to follow.
Biology at MIT.
Oh, that's true.
we're
Yeah, that's
weird.
right. It's life sciences. Whatever. Approximate numerical contours. So I'm delighted to see you all here. This is an inaugural event, so it's a bit of an experiment, and you all are helping us actually figure this thing out. But we have a new speaker series that's called the Innovation Frontier Insider Stories. And the series is part of MIT HEALS, the Institute's Health and Life Sciences Collaborative. And what this series is going to focus on is how lab discoveries in the life sciences translate into world and life-changing products. And I would say, while we pour our lives into the research domain, I think all of us have our ears tuned pretty closely to what the products of that research happen to do or can do out in the world. So some of you may not know who I am. I'm Susan Hockfield, a member of MIT's Koch Institute for Integrative Cancer Research. I served as MIT president from 2004 to 2012. And it was a period of really rapid convergence of the life sciences with engineering and medicine. And from where I sit, I'm a little biased about this because I think this is one of the most exciting frontiers in the world of innovation. But we're in the midst of that convergence driving many of the technologies that we're going to highlight and showcase in this series. So through the series, we plan to have an event, roughly once a month. Our speakers are going to be CEOs and company founders, and they're going to bring their insights to us as far as their experience in the early stages of biotech companies. So the goal, or the purpose, or I'd say the structure of these, is that we want to engage you. Audience participation involvement is absolutely central to the way we've set this thing up. So, and this is different from a standard seminar format. Your questions and your curiosity are going to drive the direction of our hour together. I'm going to kick things off with a very short conversation with our speakers. I'll introduce them in a minute. And then we're going to devote the majority of our time together for your Q&A. So I cannot be more enthusiastic about encouraging you to join in with your curiosity and your thoughts. So before we begin, I would like to get a sense of who's here. So I'm going to do a little survey. Raise your hand if you are students. So we get a sense of who's here. Great. And who among you are students and everyone are in STEM disciplines? Science, technology, engineering. Okay, good. Anyone here from humanities? Yeah, I know. Humanities or from the Sloan School? No, it is a long walk over. Yeah. Okay, we'll see what happens as we move along. But I have to assure you, although you now have declared that you all have a life sciences background, it probably is not necessary to follow today's story. You'll get more of it by having that background. But what we're really focusing is what it takes to turn a really bold idea into an enterprise that saves lives. So I'm delighted to see you all join us for our, shall we say, our starting, at the starting line. So it's a real delight to start off the series with Alnylam. Probably you have heard that name before. It's a now iconic company. It was founded in 2002 by scientist Phil Sharp, scientist and panelist, Paul Schimmel, Thomas Tuchel, Philip Seymour, and David Bartel, along with their venture capital partner, Christoph Westphal. And their goal was to commercialize what was a brand new biological entity, RNA interference, and to see how RNA interference could be turned into a therapeutic modality. So it's a real privilege to host two of the central players in Alnylam's founding, Phil Sharp, MIT Institute professor, and Christoph Westphal, co-founder and general partner of Longwood Fund. Yes? Yeah. Got that right? Okay. Well, you can correct me whenever we go off the rails here. And they're here to answer your questions about their Alnylam adventure, along with, if you care to ask them about it, the current and future state of the biotech industry. So I'm going to kick things off with about 15 minutes of conversation just among the three of us. And then the remainder of our hour together will be driven by your questions. Your curiosity will determine the direction of the conversation and the insights that we'll gain from our time together. So this is a kind of a seminar that gives you a lot of responsibility for what you learn from it. So let's get started. Phil and Christoph. So in 2002, the discoveries on which Alnylam was based were very new. And I'm just curious about how you decided that those discoveries were ready to move out of the lab into the marketplace.
Why don't you go first, Phil?
So I'll emphasize the discoveries, and there were a series of them that led to the decision that there might be a new modality that could be used to treat a wide range of diseases. And the first of those discoveries was the work of Andy Farr and Craig Mello, who discovered that you could add double-strand RNA to the food of a worm, C. elegans, and if that double-strand RNA was from a specific gene, it would silence the gene. And that was stunning because it was a new activity for double-strand RNA. We knew double-strand RNA stimulated innate immune responses, interferons. And we knew that there were enzymes that attacked it when it was in the cell. But we had no idea that there was a pathway from double-strand RNA to silencing the gene. So that was in 1998, the fall of 98. And Andy Farr was my grad student. He was here at MIT. And another grad student, that's all right, I'll get his name in a moment, showed that it was active in Drosophila embryos. You could inject double-strand RNA and silence a gene. So those two papers were in the literature. I wrote a little perspective saying if this could work in mammalian cells, it would change everything because we have then a system to take and by simple design a double-strand RNA silence genes. But Tom Tuschl, Phil Zamore, David Bartel, we decided as a group to see if we could develop a biochemical system to understand this process. And Tuschl and Zamore set up the system. Zamore had the extracts, Tuschl had the Drosophila extracts, Tuschl had double-strand RNA. And the first experiment gave a little signal. The second experiment, it was clearly there. The controls, it was clearly there. So we had a biochemical system in Drosophila, and that continued to be worked on. And that led to what is called in Alnylam Tuschl 1 patent. So this was 99, the end of 99, 2000, we knew a Drosophila extract generated small RNAs. Tuschl leaves for Germany because he has a job there. Phil Zamore leaves for U of Mass because he has a job more. They continue the work. David continues the work here, Bartel, and I continue to talk. And so we, by the end of that year, particularly Tuschl cloned RNA from that extract, discovered this siRNA structure, a 21 nucleotide double-strand RNA that when added to a cell silenced the gene. But he did that experiment in human cells. So then there was another patent called Tuschl 2. So those series of discoveries led us as a group to say, we have a technique that if I know the sequence of the gene And by the way, 2001, they published the first draft sequence of the human genome. And we know a disease, if we could make this work in a patient, we have a therapeutic modality that could potentially everything, and this is all, hypothetical at that stage, we could potentially treat a wide range of diseases. So the question was, how do you make this happen? And so it was terribly new, and the thing you want is first mover advantage. So the hope that we could make this a therapeutic modality, and the idea that we could be first mover was motivating. That's where it was at.
So, Christoph, how did you get into partnership with these biological lunatics?
Yeah, with a bunch of biochemists for whom human cells was something very different. So I had done my MD PhD at Harvard and then went to McKinsey to learn business for a couple of years and then started a venture fund in Boston. And the first company I founded as CEO was Momenta with Bob Langer, where we got a couple of drugs approved and J&J bought it. And I had just finished up that gig, getting it going for the first couple of years, and I was visiting Phil in his lab with some stupid idea I had, and he said, forget that, look at this Tushel Nature paper. And I looked at the Tushel Nature paper, I was like, wow, that's pretty cool. So I love really cool science and technology and great human beings and great scientists, and my goal is to do translational medicine, take science and turn it into medicines. And so we decided to start Alnylam together. So that's how I got involved.
So for both of you, so what didn't you know that you needed to know? Both of you had experience in this domain before. So were there new discoveries en route to Alnylam?
Maybe I can go first. So I wasn't old enough and jaded enough to realize that whenever you talk to a very excitable scientist like Phil Sharp or Bob Langer, everything sounds perfect and it's going to go really quickly. I've learned over the years it takes about 10 to 20 years and about $2 or $3 billion to go from their ideas to a drug, if you're lucky. But I knew that if you worked with great scientists, with great science, with great startup teams, good financial people, like us, and organizing it, and you had the patents, and pharma was interested, it was worth the long-term, very, very risky bet. And then I learned, but I didn't know yet, the most important thing and the most important contributor is not Phil and it's not me, but it's ultimately a world-class team that's going to spend 20 years building a great company, all the great doctors and pharmacologists. And we had a lot of things still to discover in RNAi to turn it into a drug. And neither of us discovered those. Other amazing people at Alnylam did. So I learned all that.
Wow. So one of the questions that has always been in my mind is, what did you, thinking back, what were the things you learned that were most important in facilitating the progress that you've just described? So, what are the things that, people should be focused on in terms of learning about how not just biology works, but how the world works?
Phil, why don't you go first?
Okay, so I had learned some things from my previous experience in Biogen. That the CEO and the company team was a make or break issue. You can have the right science and the wrong CEO and it won't work. If you have the right CEO and the wrong science, they might be able to create or find the right science. And the second issue is, particularly in pharmaceuticals, is the ability to deal with pharmaceutical companies and the time that it takes to develop a product with intellectual property. So that was the two issues. Thinking about what I really didn't appreciate how difficult it was going to be. The question was delivery. delivery. So we had the first mover advantage. We went to Millennium and made a pitch to Millennium as a company, not because I particularly thought they were going to invest in us. They couldn't because they were at a stage where they needed money. But we excited a lot of people, and that was good. And then the company, the delivery issue came front and center. We invited Bob Langer to join the Scientific Advisory Board, who brought an enormous amount of knowledge in that regard. But it took us 8 to 10 years to get lipid nanoparticles to be effective in delivering RNA. And then, but they would work, and they were actually, that work was critical for the whole response to COVID. It was, in fact, the lipid nanoparticles are going to open the door to new cancer treatments with cancer vaccines and CAR T cells. I think it's going to be the next renaissance in cancer therapy. But using lipid nanoparticles, which was their first proved product for Alnylam, took 16 years and $3 to $5 billion to get to that point. But the lipid nanoparticles had to be delivered intravenously. The company, that's a very expensive, awkward way of treating patients. You'd have to do it, in several weeks. We developed GalNAc. They developed GalNAc, where you can sub-Q inject yourself. The RNA is distributed and then is taken up preferentially by the liver and silences the gene. That became a widely available therapeutic. So over that time to now, I think there's 7 approved products and clinical trials underway for many more. And it's been a very broad platform. And the reason we were able to be first movers, define the field, companies were comfortable dealing with us because we had intellectual property that they could say, well, if something happened, we have access to the market. And that gave us this advantage of being able to raise money and to be able to see it through to create these new delivery modalities and to then get it approved in patients.
So I'm going to just ask one more question. I have many more I could ask, but I really want to hear from our audience. I think, one of the things I wonder about is how has the relationship between the academy and industry evolved from what I have to say was kind of frank opposition for many years into, for some places, a pretty strong embrace of our partnerships with industry?
So when I finished my MD PhD and I told them I was going to go and start companies, they thought it was like the worst thing in the world ever. But my medical school now is much more open. MIT's always been a little bit more open, but Harvard, I think, was horrific at translation. And so most of the companies I've started, actually, even in the last 25 years, are MIT companies. I've started 7 companies with, Phil and Bob Langer and Tom Maniatis, 3 of them over 10 billion sustained market cap and multiple approved drugs. And 7 of them I took public. Most of them are MIT, so I still think MIT is better than Harvard at it. It's like
Stop.
I like practical engineers, and so I went to the wrong school somehow. But Harvard's gotten better, and I think all of us understand we're part of a continuum, right? I mean, what do you all want to do? You want to discover important things. You want to help patients, right? That's what I want to do. You're not going to do it alone. I'm not going to do it alone. You're part of the solution. I'm part of the solution. So we have to work together to get medicines approved, and I'm really glad to see that that's much more common.
Let me open to the audience because I'm guessing that some of you have kind of spring-loaded arms to stand up for questions. Please. Tell us who you are. Hold on. Mic coming. Thank you.
I want to continue that conversation and learn more about MIT's policy regarding faculty running their own business on the side in terms of like who owns the pattern, how do you share profits, what's the agreement on time allocation, and other legal issues. Thank you so much.
Hey, Phil.
So there was I can speak best from the Alnylam time. All these patents that I had talked about were collectively pooled and licensed in two ways. This was all through the TLO, Technology Licensing Office, that they were licensed to laboratory supply for semi-exclusive, not semi, but licensed, and licensed therapeutically. One in Europe, one in US, but then that was consolidated in Biopharm. MIT took their proportion of the ownership in the company. That was in the TLO office, and there was royalties on the income from any sales. I, as a faculty member, I participated in the royalties. Those royalties and internal assets were, at that stage, I think a third went to MIT, a third went to the department, and a third was split among the discovering scientists. The rules then was if you were an MIT faculty member, you couldn't be a principal in a firm. I think that's evolved in some ways because of the whole AI social media world, because things are much quicker there. Okay, so that was the deal. I've been at MIT. Started when Biogen started. There was no consulting out of biology. There was a lot out of engineering. But MIT's had, very workable but known rules about what was appropriate for faculty, with the overarching rule that your primary interest was at MIT.
One of the things that continues to puzzle me is that MIT really has set out the standards for how to do this kind of cooperative work in ways that keep the academy strong, allow industry to grow. And the thing that has puzzled me is we have a model. It's largely not been adopted. I ran into some kind of tough sledding at Yale around these issues. I'm thinking, wait a minute, my friends at MIT can do this like in a nanosecond, and I couldn't make it happen at Yale. So why is it? Why is it so hard to copy the model, which we've all enjoyed? Why doesn't it work in other places?
In my experience, at a place like Harvard, where I went to school, or even Yale, they have a medical school. So that's a whole other added level of complexity, because you definitely want to separate any inventor from an intuition that they might want to try their invention in patients. So there's a little bit of added complexity with universities that have a big medical school. But then the separate thing, I think, is MIT is practical engineers who want to make the world a better place. There's less ego, honestly. And it's part of like, hey, if your thing is important, someone's going to create a company and make a product. That's a great thing. In a place like Harvard, in my experience, it's a little bit more ivory tower.
Interesting. Audience, other questions? Down here, please. Whatever. We'll get to everyone.
Yeah.
Well, I can say that. Thank you. We'll get the microphone moving. Thank you.
So I have a question. It's been quite a while. How do you think the funding, from your standpoint, how has the environment changed? Are you more risk averse now I mean, I hear you mention the same names over and over, and that's just a fact. I mean, when you hear Phil Sharp or Baumlanger or Edelman or whatever, you hear them over and over again. How much does that influence your decisions? And would you say would it take more for you to invest in something like an Alnylam now, today? Would you think twice about doing that? It's been a long road. So.
I think you asked 3 questions,
Sorry.
actually. So, no, that's good. One question is, we've just gone through a brutal 4-year downturn in biotech. Everybody has completely lost their shirts investing in biotech. And so that has made it very difficult. I mean, I love it, and I still do it, and I just raised another fund. But my guess is like every downturn, 2000, 2001, 2009, 10, 11, a third of the funds will be gone. All the lab space is available. There's a lot of unemployed people. So actually now is the best time to start a company. Like Alnylam, we started in 2002. It was a brutal period. So the first thing is it's a tough period now, but that's the period where all of us should be starting companies. The second question I think you asked is related to the same people. I mean, it's kind of a weird thing, right? Like I have this thing of like, if someone has a bunch of Nature Science Cell papers, like maybe they're likely to have another Nature Science Cell paper. For me, if someone knows how to start a company and what's involved, maybe it's just going to be easier and more straightforward to start it with them. So I think we all in life, like you want to go when you have neurosurgery to the best doc who's had good results in the past, right? So there's it's not just that people are famous. That would be the wrong reason to work with them. But hopefully they've been successful because they've learned lessons and they've made mistakes. But there's like 10 or 15 or 20 or 30 amazing young people who I'd absolutely start a company with now. And the third question, I hope I would start and invest in Alnylam now. Yes, I hope I would do it again. But Phil, do you want to add to that?
Can I? Oh, yes, I have a mic. So, yes, I would want to add that if you got a great idea, and in my case, I've always sort of, what I do is pretty basic science. And I really wanted to be able to use that understanding of basic science to make a difference.
Yeah.
If you can, if you have an idea and it's really interesting, collaborate. Find somebody, pitch to them, see if they are interested in helping you. And if you can get people to buy in, then you'll have a team of people who can approach someone like Longwood or others and say, we have various dimensions of understanding this problem and the process, and we're ready to somebody is going to fund us and we're going to do it.
Yeah.
And that makes a great deal of difference. If you want to stand alone, it's really a challenging, brutal. It's always been that way.
Yeah.
I'm saying it's something to think about.
By the way, one added thing I want to tell you. So when you've got your cool result, you want to start a company, you meet like a bunch of people like me. They all sound like they're reasonably smart, and a lot of them are BSers. Ask them what companies they've started that have been successful, that made drugs, that created value. I think you should work with those kind of people. Like, just like I maybe would work with Phil again, you should pick. It's not only the science. In fact, if you asked Phil and me, he said it very clearly. It's not actually the original science. It's the people you get into the company. So the first person you want to work with, like, what are the companies you started? Were they successful? That's a pretty good predictor if they can do it with you. So that would be my strong plea to you.
Great. Next, yeah.
Yeah, thank you so much. I'm Diana Grass. I'm a 4-year PhD student here at MIT in HSD, so Harvard too.
Oh, are you doing an MD as well or not?
The PhD.
Okay,
PhD.
well, just think about it. You might do an MD too. Think about it.
Thank you. So you mentioned the importance of the team, but as scientists, we are not always aware of this business part. So what are the traits that you choose? Like, what are you looking for when you're thinking about like a CEO or a CTO or people that will work with you if you are like thinking about creating a company? So could you be more like detailed on what traits do you see? And if it is not just the science, what are the other traits that will let you know this could be a successful partnership?
Well, for me, it's important that the team has as a major leader, probably the CEO, someone who understands the science and the pharmaceutical development challenges and the medical opportunities. And as Christoph commented, if they've been there, done that, that's the best person, where they can say they've seen it. And the same in terms of business. I have gained an enormous respect for good business people. They pay their dues in the pits, working in various environments. You heard Christoph McKinsey, where they really did learn things and how other people solve problems and how they've solved problems. You know, in Biogen, it was Jim Denson. He came in, he was a long-term entrepreneur, business leader. He shucked the company, split it, and reduced the size in factor of 2 and focused it. And we survived because of that. So, you know, those aspects, learn, make a network. You're historically, it's called a Rolodex. But your network of people that you can learn from and share ideas with, and they could possibly introduce you to someone else who then could be beneficial in funding or advising or opening other doors, is extraordinarily important. So those are and then it's always been there, done that, is really another standard that I think is terribly important.
Yeah, I have 15 MDs and PhDs. They're all like Harvard or MIT, who work at Longwood Fund, and they're all incredibly smart. And for me, like, what's going to turn them into a great venture capitalist or someone who can start companies is we get all these amazing people like you. They come in and pitch, and then I always ask these MDs or PhDs who work with us, like, okay, what did you think about it? And of course, all of us are going to gravitate to, is the science great? Yes, it has to be. Are the scientists behind it great? Yes. Or, are the patents good? But at the end of the day, the key thing is, what about those people? Is that individual who's the CEO of that company going to walk through walls and make it happen? So the people you work with, the people you pick, are at least and more important than the original science and technology. And I want to see for venture capitalists that sense of people who you bet on, right? Under promise, over deliver type people versus flowery promise the sun and moons. Like Jim Vincent saved Biogen because he shut down Geneva, fired half the people, focused the company. Otherwise, Biogen would be dead.
We wouldn't have saw it.
So that's what you need in these companies. And there's a lot of companies going out of business right now with poor leaders, but there's some amazing companies that are going to thrive. That are getting through
But I
this
want to
period.
say one other thing about Jim was very important. Just setting the context, Biogen went public at 23 a share. When he came into the company, we were at 4. You think your stockholders are happy? And we were, had enough cash to run the company for a year, but that was it. Yeah. And not only did he do this, but he was able to sell on Wall Street and other investors that I'm in charge and I will make this happen. So there's a credit that comes in the external world, which academic scientists have almost no influence on, that, I know what I'm doing. I'm betting my career on this activity. You should join me. And he had been successful 2 or 3 times. There were people who came in and invested just because his name was on the title. That's a business.
Let me just ask, so was that the only valley of death that you all faced, or were there other
Well,
moments when?
Alnylam is sort of
We went public at 650. We raised $30 million in our IPO, barely got it done, and traded down to $3 a share. And we're literally, for 10 to 15 years, almost worthless. And John Maraganore kept it
Well,
going
I
with you
wouldn't
guys.
say worthless, but had a very low price, shall we say.
So, and we had a bunch of pharma deals that kind of kept us going. And
Yeah.
in 2011 and 12, nobody believed anymore in RNA interference. Every pharma company shut it down. It was like...
Sorry, so how long was the journey between the lab discovery and a drug?
About 15 years and $3 billion.
No more.
Maybe 16.
We became profitable in 20...
Well, he's talking about profitable. First drug approval. was
First
at
drug
15.
approval was in 2016.
Yeah, so 14, 15 years.
Okay, stamina. Who is the mic now? Thank you. I mean, one short question is, how do you keep the money flowing if it takes 15 years?
Well,
Well, I have 5 kids, and I tell my kids, my job is I talk to 100 people every day about my idea, and 99 people tell me it's a really stupid idea. And that was a good day, because one person thought that was a good idea. That's my job. That's the kind of person you need, right? You need to just keep on going.
well, but more specifically, because I think it's you can have him, or you could have relationships with Novartis, Merck, Sanofi, and there's a couple of others in there. So those were all signed with some upfront money to support the science, giving rights to products that are being developed, partial ownership in the company. And then there was a loan. I mean, there was a IPO, a subsequent second marketing, a debt that then was built on possible potential royalties in the future. So, I mean, it was a mosaic of avenues that was used to be able to get the technology in line, get in patients. It's one thing to talk about everything that works in a lab, but you have to get in patients as fast as possible.
Yeah.
And see, what is real, and then persist and persist. And here, persistence was, if we can get the first one done, the second one will be easy, and the third will be easier, and the fourth will be easier. And we can change this technology because we can go to other organs and we can do other things with this technology. So it was, if you could ever get there, it was going to be sweet. And that was the pitch.
Who is the mic now?
Yeah. Hello, I'm Nishat. I'm a fourth-year PhD candidate in chemical engineering.
Cool.
My question is, so like coming from a background where you've had scientific training and then moving into like this business translation space, like how did you have to retrain your thinking or like manage your own biases? Because when you come from a scientific background, you have a certain way of thinking, but in the business translation world, everything's moving fast. You got to make quick decisions. So how did you have to shift your mindset, or did you even have to do it? So yeah, that's my question.
That's such a great question. Thank you. Every world has its own peculiar idiosyncrasies. So there's the medical world where it's like a lot of memorization and rote and algorithms. Then there's the science world you live in where the primal goal is the creation of knowledge and discovery. And so every world has its belief of a hierarchy of values, right? But actually, if we just understand that every world, then not one world is better than the other world or worse. And actually, my view in science, that the greatest good in humanity is discovery. That's just our belief in our little world. When you get into the business world, it's about generating value, right? So business people and investors, that's what they care about. So yes, each of these worlds, you have to retrain yourself. And the way to make a medicine is to start from the discovery primacy world, get through the translational medicine where it's all about, oh, can we help the patient? By the way, at the end of that whole path, there's a Lilly, or now an Alnylam, but a Lilly that's selling $50 billion a year of GLP-1. And that company is essentially run by marketing people. So we just have to try and remember that we're just small parts along that value chain. We're just trying to integrate it in a biotech. Take it from discovery to get the drug approved. And now Alnylam marketing. I don't know if that answers your question.
I will say the skills you've learned in doing basic science, be critical,
Yeah.
understand what's all sorts of validity that you can really depend on, are very important in all that process. But the ability to select and read people and to make networking connections that allow you, you're going to have to practice as a team. You've got to learn how to work with teams, lead teams, not be that you can be part of a team and influence a team without, being the head of the team. But you need to work with a team and continue to respect and expand your knowledge of what they do and how they do it and what really critical touch points each person brings to an endeavor. And it's that ability to work in an interactive mode, which you learn in business. If you're selling widgets on a corner, you understand how to respect that customer and relate to them and try to solve their need or want. And that's just part of what you have to do.
Where's the mic now? Yes, go ahead. Can we get the mic down here, please?
Hi, my name is Kevin, postdoc, currently also co-founding a company. I think my first question would be, how do you pick your first indication? Like if you have a cool technology, how do you choose which disease you want to go first? Is it market size driven or is it like science driven? And how do you balance? Because if you pick a rare disease, it's easier to get into clinic, but then your market size will be lower. And then when you pitch to investor, you have to balance, right? And second question is, now all the investors are very interested in AI. So for company who doesn't have an AI, like we just have a technology, can do a treatment. So how to pitch, any advice?
So I think Phil said it correctly. The key for us, you all and me in my role, is to get to human efficacy data as quickly as possible. Until you can show in a human that your pharmaceutical has a beneficial effect and not too many side effects, you really don't know what you have. So generally the model for biotech is to go for severe indications, no matter how small, and as quickly as possible show that your drug works. If you're lucky, like Alnylam was, and that is also luck, you happen to think you're working on something that may be somewhat rare, like ATTR cardiomyopathy, and then it turns out, actually a lot of patients have this. So you get it approved, and then it's a massive market, and then you're a very valuable company. But you basically got to get to human efficacy as quickly as possible. The second thing, I think if you have a really cool science and a really important drug, it's fine. Like following the fads and AI this and everything, I prefer to skate where the puck isn't. So I don't know, Phil, do you want to add to that?
Well, I agree. Particularly if you're a small company, you have to focus. And if you try media, that's another world. And it's a very different, the media world works in a 2 to 3 year window. If you can't get to some element of profitability in that sort of space, or at least high credentialed, you're typically find it hard to fund things. Pharmaceuticals have this problem. You got to get into patients, prove patients are benefiting from it and safe, and that's a 7, 8, 10-year window. So I think they're very different worlds. They've been as different since I've been in the field. Now, I think the point where they're going to come together is in healthcare delivery and re-engineering healthcare in terms of patient access, educating patients, bringing them in, supporting physicians and physician assistants, treating patients. I think medicine has to be re-engineered in this country. And I think that's where there's going to be the most intimate connection between AI and medicine, in my opinion. We're going to be able to discover drugs more rapidly with AI. Every company, I'm consulting for a couple of companies now, all of them are using AI in the back room to accelerate their discovery. But it's almost the type of AI you could use in a laboratory here at MIT if you had a big enough budget. So it's accelerating discovery, and you have to understand how you can do that because it makes you more efficient, more likely to have on-goal activity. But it's not a 3-year AI window.
So we are focused a little bit more on the biology, which is understandable, but I wonder that as you started the company, were there patent and commercialization issues that you needed to address?
Well, at the beginning, there were several places where IP was held. It was MIT, that was Lena Nelson, UMass, Max Planck. There was also other patents not related to the human RNAi, and so the key was to get most of that IP. We wanted to get all of it. We almost got all of it, but then there was a little part of the UMass patent that we didn't get. So we had to get those pieces together. The crazy thing, I don't even know if you know this, Phil, those original patents, those royalties didn't pay out, as far as I understand. It was too long to approval of the drug. Yeah. So MIT made money on the equity, but not on the patents. But the patents allowed us to do deals with several major pharma companies, which kept us alive and allowed us to develop the drug.
And there was all sorts of potential litigation through all of this.
Yes.
So if you are trying to protect a fundamental intellectual property portfolio, you're going to have a budget to deal with litigation. It's the big boys will just run you into the ground if they think you can't do it. Yeah.
We have time for maybe 1 or 2 quick questions. Anyone? Go ahead. Let's start. Yeah.
Hi, I'm a PhD student at MIT in the middle of my MD PhD also. And I had a question about kind of the consolidation of therapeutic production. Do you see the future of biotech kind of therapeutics being consolidated into these major companies and these major pharmaceutical companies owning a lot of the intellectual property in this space, or do you see there's room for small companies? And how does N-of-1 therapy production fit into that?
Got it. First of all, I want to know, where did you get the cool sweatshirt? I've been looking for a Harvard Medical School sweatshirt. I can't find it.
I made it myself.
You made it yourself? Yeah, I have my reunion, and I'm like, you can't find a HMS sweatshirt. Okay. Well, maybe you can make me one someday.
Well, go into business.
Yeah. I think that innovation happens with incredibly talented, young, smart small groups of people. And so I'm never afraid that the innovation in biotech or in any space in Silicon Valley will always come from small, extraordinarily creative groups of people. And so ultimately, they all get hoovered up, most of them, by pharma. I hope Alnylam stays independent forever. I fear maybe someday it won't be independent. But I think that the general rule is almost all innovation is coming from little companies in biotech. I do think, to get to your second question of therapeutic, sticking things, one thing that's changing right now is that 50% of medicines being licensed from pharma is now coming from China. So that's a big change. That's because China is faster and cheaper. And I don't know, maybe they're smarter, I don't know. But so that's one thing I'm like, wow, that would be a shame if we at MIT and Harvard weren't still the epicenter of innovation in biotech. And so we got to figure out how to get into the clinic quicker. I think the problem isn't that you're not smart and creative, it's that it takes too much time and it's too expensive to do the initial human studies in the United States. And hopefully we can fix that, and I know FDA is working on it. Phil, do you want to answer that question a different way?
Manufacturing in pharmaceuticals is an interesting issue. If you look at the price of a drug, the manufacturing goods is a very small percent. So what do you have to, you have to be nimble, you have to be able to identify and be able to make new compounds or new substance. RNAi is 21 nucleotide oligonucleotide. It's a very specific, high informational molecule. It's more expensive to make. But bringing that expense down will make it a more possible generic market in the world where it will be available. So there's a great challenge there to do that. I also think that there's going to be a gradual evolution of some major hospital systems setting up their own experimental production systems for testing cancer vaccines and CAR T-cell RNA delivery. I think RNA will be the model for that. And that's going to accelerate a local production. I know several major hospitals in cancer are doing that, setting it up now. So how all that's going to evolve, I would pay a lot of attention to high-tech, small, nimble, AI-driven production facilities that can be plopped into various environments.
You have Mike? You have been asking for it. I apologize for not.
So I imagine that Phil and Christoph, you both have had so many ideas over the years, and Alnylam obviously was a successful one. When you look at your other ideas, do you think like that one would have been good too, I just didn't have time to do everything? Or do you say like some of my ideas I can see, like I chose not to pursue that one because I'm curious about why that would be?
Maybe I can, as a venture person and a founder. So a typical year, should know the exact number, but it's, 2,000 companies come and pitch us. And my guess is that 1 to 5% of them will work out, maybe. So hopefully we're pretty good at picking out which 1 to 5% will work out. But I think the hard thing to remember for all of us in this room is there are so many ideas and so many companies, and the vast majority will never work. Is that shocking to you or not? Okay, so that as a backdrop, it's like, whoa. And then how do you get a little bit of pattern recognition about the things that maybe could work? First of all, when Phil tells me an experiment worked, I believe him. Unfortunately, that's not true for every academic lab in the world for me. So first of all, is the science credible and the person is credible? Do they actually want to do the right thing? I knew this wasn't about Phil's ego. He actually wanted to make a medicine, make the world a better place. That's a really great thing. The next thing is, could he work with other people and attract other people? Absolutely. I mean, for those of you who went to medical school, you know, DSM-5 now, I guess it's called the Axis 2 personality disorders. I have that on my shelf in my office because all of us have some aspects of certain personality disorders. You don't want to have people have 7 or more of the criteria of narcissistic personality disorder or borderline. We all know people who are like that. Those people will destroy your company. At every point, Phil and I were joking, he got fired from the board of Alnylam, I got fired as CEO of Alnylam. We're all gonna get fired at some point. The goal is Alnylam needs to be successful, and the drug needs to get there. Our ego needs to be left outside in these efforts. So, sorry that I went on and on, but those are some of the learnings I've had.
Answering the part of your question about what you see, a lot of things that are interesting that are potentially useful, and hopefully you contribute in your lab to those in a direct or indirect way. But I mean, my preference is if it's really fundamental science and it has broad application, it could really change large portions of medical care. Okay, I'm willing to spend the next 15 years trying to make that happen. All right, I'm going to have to, my primary commitment is to MIT as a faculty member, but I'm going to spend the other part of my time and my intellectual reading and my talking and my engagement with people making this work. Right? And I don't do that. If I see that between me and that patient is Lily, right? Because I respect what Lily does. They have bright people. And if you have to move through them, you're not going to be successful.
Yeah, I mean, that's a good point. 15 years, 20% of your time. For me, it's 90% of my time for 2 to 6 years. Like, I'm not going to live that long. Like, what do I want to spend my time on 80 hours a week? You want those kind of people who are like selecting towards that. And you have to decide what you want to spend your time on.
Unfortunately, our time is up. I've got another 2 or 3 questions in my head, but we won't be able to get to them. Thank you for your attention. Thank you for your questions. And most importantly, Phil, Christoph, thank you so much for giving us insights into the inner workings of this fabulous, fabulous industry. Thank you.
Thank you.